Good afternoon, everybody. Thanks for coming. For those of you I don't know, I'm Mike Clegg. I'm Dean of the school, and want to welcome you to this Dean's Lecture. As you know, we hold these for special events, primarily for people who have received the honor of being promoted or appointed at the level of professor at the school. And as I always say, to be appointed to the faculty here is a big deal. But to be promoted to professor is really remarkable, and it's a decision made by peers around the world, not necessarily by us. You know, we have a committee of faculty who review the letters that are sent in. So it's a wonderful affirmation of one's accomplishments to be appointed professor. And as you all know, we're here to recognize David. And you all, I'm always in this position of introducing the person who's promoted when probably everybody in the room knows him and his work, or her and her work, better than I do, but I'm going to do my best. Okay, so David graduated with a PhD in evolutionary biology and ecology from Princeton, and then stayed at Princeton and got a master's in the same field, and then got a master's in mathematics from Brigham Young University, and then did a postdoctoral fellowship at Maryland in the Department of Biology in the biology of small populations. I don't think that means short populations. That means small number of populations. And then was at the University of Maryland School of Medicine for many years, and went from there to the Fogarty Center at NIH, and then to the University of Florida. And we were very fortunate that the Malaria Research Institute here was able to recruit him here. And his work, as you all know, he describes himself as a mathematical epidemiologist and disease ecologist. And he's made many contributions in a whole variety of infectious diseases, but I would say his most important contributions have been in understanding malaria, both the epidemiology of malaria, the spatial dynamics, the transmission, and the mapping. And I'm sure he's going to talk a little bit about that in his work today. He has from my interactions with David, he has a unique ability to look at problems in a different way and to use quantitative methods to help us understand those problems. And he's going to be talking about the theory for malaria eradication today, which is something he's worked on. And I don't know if he'll get into the 80-20 rule. Everybody in medicine knows the 80-20 rule, or there's an 80-20 rule for everything, that, you know, like 80% of the costs are incurred by 20% of the people, or, you know, 80% of the alcohol is drank by 20% of the people. Well, it turns out that there is an 80-20 rule. Are you going to talk about the 80-20 rule? So I won't steal it, but I'll just say that I'm one of the 20, I think. Okay, so please join me in welcoming David.
Thanks, Mike. I'm delighted to be here, and it's nice to see so many faces out there. I know, nice to have the crowd back from Life of Riley. So global eradication now defines the long-term goal for malaria, and it actually has since 1955, when the World Health Congress voted to make it official policy of the world. But that sort of 1955 vote launched the Global Malaria Eradication Program that lasted about 14 years and then failed. And in the wake of that, there was no enthusiasm for malaria eradication until in the late 1990s, the problems associated with chloroquine resistance put malaria back on the public health map, and some leaders of African countries met in Abuja and had a conference and a discussion. And this rekindled interest in malaria control, leading up to this event, which was the Malaria Forum in the fall of 2007, when Melinda and Bill Gates decided to devote the full resources, or they were going to put the resources of their very well-endowed organization behind malaria eradication. And after they said that they were committed to eradication, Margaret Chan stood up and committed the World Health Organization to eradication again, and so on. And a consensus formed over the next few months actually, that the world was turning to eradication again. So it now defines the official, Rollback Malaria has agreed this is the official policy. And in the wake of the announcement, there were a number of documents produced. In fact, there was a whole lot of ink that has been produced. And right now, this is ink sort of reviving the discussions about the theory supporting eradication. rather than, say, doing things, because we're still very early in the game. And so if you think about this, and I'm probably preaching to the converted, but it's worth saying that everything we do in science and policy works on some sort of model, either a conceptual model, a statistical model, a mechanistic model. And the models and the theory are actually very useful, because that's how we translate a success done in one field to a success in another. It's not all on data, it's on an idea that something works and the evidence that it works, and that's how we extrapolate from one thing to another. So I'm going to talk about theory for malaria eradication. So malaria is a disease that's caused by infection with a parasite, and that parasite is transmitted by a mosquito. And when it comes right down to it, it's a very complex life cycle, and I'm not telling you anything you don't know, but the parasite undergoes various stages of development, and it actually undergoes sexual, sex happens in the mosquito, so the mosquito is called the definitive host. Maybe true for biologists, we don't really care about mosquitoes getting sick that much. But there are a number of very interesting questions, scientific questions, that occur as the parasite develops from gametocyte to sporozoite in the mosquito, and then from sporozoite back to gametocyte in the human host, including the development of immunity in the mosquito and in the human. But if we're going to understand what's going on overall, we have to go beyond what's happening inside the host and to think about transmission, which is focused on this act of blood feeding. So the mosquitoes take blood, not for the sake of propagating the parasites, but because that's how they make eggs. So they provision blood into eggs. And the intensity of transmission is determined by the frequency of blood feeding, and this is described by various things. And as we move from this focal event of the blood feeding up, we start to focus on other phenomena, ecology and behavior of mosquitoes, dispersion of the parasites in infected mosquitoes and infected humans, the epidemiology and health and disease of malaria in humans. So as we try to control it, we think about all the things we can do for vector control, long-lasting insecticide-treated nets, indoor residual spraying, larval source management, all the things we could do in the humans. Mostly now it's give them drugs, hopefully someday soon, vaccines. And we can measure those effects on transmission. We worry about insecticide resistance, we worry about drug resistance. We should also be as concerned about the sustainable financing, so donor resistance, if you will. And as we move up into even bigger scales, we worry about the different vectors that transmit in different biogeographical zones. Human mobility as humans move the parasite all throughout the world. National malaria policies, which are a hodgepodge of drug and vector control policies. At the very biggest scales, we have the global burden of malaria on the one hand, global change, and this big issue of global malaria eradication. So instead of thinking about the parasite life cycle, we think about a different picture that sort of moves from things that are happening close by to things that are moving at an even bigger and bigger scale. I want you, that yellow at the top, Derek Cummings asked me to highlight his name, so I did. So. This paper just came out yesterday, by the way, with these graphics in it. So today I'm going to try to talk about the theory for eradication in terms of that scaling problem. And I'm going to start with the theory of transmission. So how do we understand transmission in populations? How do we understand dispersion of the parasite? We're going to talk about a theory of control. How do we understand all the interventions that we do to suppress transmission and cure disease? And then we're going to get into some of the nuts and bolts of an idea called spatially progressive elimination and what's behind it. And then I'm going to wrap up. So a theory of transmission rests on the idea of reproductive numbers. Now, you probably know it from other infectious diseases or perhaps from malaria, but this was the first reproductive number for any infectious disease. It was named for malaria. So MacDonald was clearly aware of Lotka's work. Lotka was a demographer who spent, in fact, quite a bit of time at Hopkins and coined the phrase a basic reproductive number to look at life tables. And MacDonald borrowed the phrase, not the terminology, and he invented this concept. So I'm going to talk about it. The biggest problem is that when we talk about diseases that are transmitted by vectors, the vector populations are what determines the intensity of transmission. And so malaria, for example, on the top you're seeing Plasmodium falciparum, on the bottom you're seeing Plasmodium vivax. So these are two of the parasites we care a lot about probably account for more than 90% of the burden of malaria across the world. And what you're seeing is the color of these is indicating something about the intensity of transmission. So the biggest pattern here is the very bright yellow and even red dots in Western Africa, northern Mozambique, and DRC. And you'll see some brighter spots if you were to look at it even closer, in Orissa in India, Papua New Guinea. And similar kinds of variability is occurring for vivax across the world, and a large fraction of what's going on here is related to the underlying mosquito populations. Ross said it this way. So Ronald Ross was the guy who discovered that mosquitoes transmit malaria, and also one of the first, in this publication, developed one of the first mathematical models describing mosquito-borne transmission. And he noted this: the infection rate varies not only in neighboring places, but even in the same place from year to year. And this, the reasons why are so important as regards to both general theory and prevention that we must endeavor to obtain clear ideas about it by careful reasoning. This careful reasoning for him was the development of mathematical models. So he started from a very crude description of the parasite life cycle. And he sort of noted that we would have to consider both humans and mosquitoes and the parasite transmission, as the parasite is moving back and forth from the human to the mosquito. And the way he did it was to make a very simple assumption that a human was either susceptible or infected, so S or I, and that's the green. At the bottom, we have the mosquito is either susceptible or infected. And the humans were becoming infected and then clearing infections and then getting infected again, so it's a sort of SIS model. And at the bottom, the mosquitoes were becoming infected and dying. And the red dots are an assumption about transmission, about how often transmission was occurring. And these were embodied in a mathematical model. This is the second model that Ross developed, and this is from a 1911 Nature publication. But the equations actually appeared before in the second edition of a 1911 book. And so we had one of the first mathematical models for any disease, and it was the second model for malaria. And then 40 years later, we finally had data and concepts from the mosquito people to actually describe what Ross had been intuitively grappling towards but not reached. And so what happened then was McDonald finally put names on things that made sense. So the feeding rate, how often do mosquitoes take a blood meal, was one of the parameters. What fraction of those bites are on humans, and so on. How long does it take for the parasite to develop in the host, so sporogony. How many mosquitoes are there per human? And all these parameters became what is known today as the Ross-McDonald model. And so not too long ago, we published this review, and sort of a historical overview of all of the ideas that went into the formation of the Ross-McDonald model, which you can trace to maturity, I would say, at about 1969, which coincided with McDonald's death and the end of the global malaria eradication program. At the end of that time, though, after McDonald died and the urgency of eradication left, the study of mosquito-borne diseases became more of an academic exercise. And over the next 40 years, literally hundreds of mosquito-borne disease models were published, and they were all heavily influenced. So this is a paper that we followed up with a systematic review of all the mosquito-borne diseases, and I honestly can't remember the number of them. I read every single one of them. It was something like 350-something models and 325 papers, and it drove me crazy. Getting it all done, you can see how long the author list was because we actually scored things. But it complicated, lots of factors involved in sort of understanding all of this. So I'm going to boil it down to just a few ideas if I can. The first one is called vectorial capacity. So for a mosquito to transmit a parasite, it has to become infected, survive long enough for the parasite to develop, and then it has to give a number of infectious bites, right? McDonald came up with a formula in the upper right-hand corner that embodies that, but I actually find it's easier to understand it in a different way. So you have mosquitoes coming up out of aquatic habitats at some rate. Scale it per person per day. And then each one of those mosquitoes is expected to give some number, of course it's variable, but they'll give some number of bites on humans. And that average number of bites that a mosquito will give over its whole lifetime, we're going to call S. And P is the probability of surviving long enough for the parasite to develop inside the mosquito to where you get these sporozoites and sporogony. So you could be infectious. So it turns out that vectorial capacity is just the product of those three numbers, and of this one, of S, twice, the number of blood meals. One of them corresponds exactly to the probability of becoming infected. The other one corresponds to the number of infectious bites it would give thereafter, and P is the probability of surviving long enough to have sporozoites after becoming infected. So vectorial capacity is the expected number of infectious bites that would eventually arise from all the mosquitoes that bite a single human, and we assume that it's perfectly infectious, and I'll tell you why in a minute, on a single day. So vectorial capacity is a really simple concept. If you could somehow watch people being bit by mosquitoes and follow them, you would get a concept that would tell you how many infections would arise from them on a single day. Well, the reason we assume perfectly infectious is because humans are variably infectious over time. So, there we go. So the companion concept is called the human transmitting capacity. So how many days effectively is a person infectious? And we sort of translate it into perfectly infectious days, even though they're spread out over a large number of them. So here's a paper we just published that came out a month ago or so. And this is based on simulations of a mathematical model. The data underlying this come from the malaria therapy infections. So it used to be that we could treat malaria with quinine, but we couldn't treat advanced stage neurosyphilis. So we would give people malaria to cure their syphilis. We did it. And the guy who invented it won a Nobel Prize. But the point was to induce a fever. So these are infections that were actually deliberately badly treated. They would give enough of the drug to stop the fever from killing you, but then they wanted that fever to stick around to make you get rid of the syphilis, right? So they were carefully monitoring these things, and we have daily parasite counts on these individuals until the parasite's clear. So we're modeling that data, and what happens then is there's a cycle in the blood where you make asexual parasites, and that keeps going until the immune system finally catches up with it. Some of those become gametocytes, and some of those gametocytes then become infectious. So some portion of these are being thrown off as these male and female forms, and then they can be transmitted. So the more sexual forms you have, the more likely you are to become infectious. And here's all the data from all those studies describing sort of gametocyte densities on the x-axis versus probability of becoming infected. Now, it's noisy. It's incredibly noisy, but there's a pattern there, and it sort of follows a sigmoidal curve. The more parasites, the more gametocytes you have, the more likely you are to infect a mosquito. And so a person, from this cloud of infections, you get this cloud of people being mean infectivity over time, and it boils down to a single number, 32 days. So we're effectively, on average, these people were fully infectious for about 32 days. Now that was partially infectious spread over up to 400 days, but that was the mean. And so we now actually, and so you can kind of actually see the schmear here of what that looks like. So some people are actually infected for 140 days, right? So there was a histogram that we generated from that. And this is all consistent with that data. So now we have two concepts. How many infectious bites would arise per day? How many days? And we take the product of these two, and we get this thing called a basic reproductive number, which is the number of humans that would be infected per human, or the number of mosquitoes per mosquito. And there's one other number, which is that some bites by infectious mosquitoes don't actually transmit parasites or sporozoites into the wound for reasons that we don't understand, but we've actually nailed down that number as well. And now we get this basic reproductive number, right? It's this vectorial capacity, something that varies from place to place because of mosquitoes, human transmitting capacity, which is the way you've defined it, because we're ignoring drugs. This human transmitting capacity is sort of a raw number that would apply everywhere. And R naught then is just the product of a lot of these parameters that describe what it takes for a mosquito or for a parasite to complete its life cycle. So it has to last some time in a human. There has to be that person gets some number of bites. Some fraction of those mosquitoes become infected. They then have to survive sporogony. They give some number of infectious bites, and some fraction of those infect a human. The product of all of that was McDonald's formula for R naught, which I've explained in this different way of the vectorial capacity times human transmitting capacity. Very well. That is, importantly enough, describing a concept called the basic reproductive number, which, when I tell you the following, will seem irrelevant. It describes transmission in a population that has no malaria, that's never had any malaria, and that has no parasites. It's actually not irrelevant, and I'm going to explain why. We use that number to tell us about how we should do malaria control, because what we actually want to know is what's happening with transmission in any real population, and we have to be able to measure transmission in those real populations. And from that, we then infer something about R naught, which tells us how to do control. So I'm going to talk for a minute about the metrics we use to measure malaria, and I put Ronald Ross's picture up here for a very specific reason. A lot of these metrics were actually either developed or improved by Ross or suggested by him. And they came right out of the mathematical modeling. Ross knew that we wanted to have a very specific measure of malaria so we could do diagnostics, for example. So he developed something called the thick film. Now, apparently this had been forgotten by everyone, and I rediscovered it, sort of writing this history. But Ross actually developed the thick film, which I didn't know. It was one of the most crazy things. He also suggested this thing called the entomological inoculation rate, which 30 years later was actually done in these studies in Sierra Leone. He also invented, he didn't actually invent, but he brought to mathematical epidemiology the concept of the force of infection in some of his 1916 and 17 publications, where he laid out the foundations of epidemiology, really. Some of the most important publications now long forgotten by many of us. And by the way, some of these issues are laid out in a very recent, this is Advances in Parasitology publication that came out 2 weeks ago or a week ago, and it lays out sort of a review of the precision, accuracy, and costs of measuring malaria transmission in these ways. So let me go into some detail. We call this first one is the parasite rate. It's actually the prevalence of malaria in a cross-section of a population. Now, if you went to most populations of the world and you took a cross-sectional survey, you would find a pattern that looked something like this. No one is born infected. In a place, they rapidly become infected up to some plateau, and they stay infected at this high rate until about age 10, where prevalence starts to decline later in life. By late in life, it's reached about a third of that value, and it, typically reaches that third of that value sometime around age 20. So this is one data set from Achenga. We have now 65 data sets that are just like it. And it should be said that this pattern is not always followed. This is the typical pattern in a place where you have constant exposure throughout life. But in places where you do get something like this, this is a pretty steady pattern. I mean, we've looked for big deviances from it. We just can't find any. So this is actually done by two methods. One is the standard thick film or light microscopy. So you're looking for parasites under a light microscope. So this is what it looks like. The other one is this rapid diagnostic test, which are increasingly in vogue today. This is I'm not going to go into detail. This is a rapid way of telling whether you have parasites. So you put a drop of blood on a piece of paper and it changes color. And that tells you. Now, they're not measuring exactly the same thing. But the data that you get from surveys with RDTs and light microscopy look remarkably similar. So we use the data interchangeably. We actually don't use PCR data to make maps like this. And this is, when I showed you this map, what I was actually showing you was the prevalence of malaria from these cross-sectional surveys, but standardized to this high level that's reached in older children, 2 to 10-year-olds. So that's what we've actually plotted. And it's using data from all kinds of studies and people in all kinds of ages. And we describe in that previous paper down here how to do the standardization. So the other metric, another metric we use frequently is called the entomological inoculation rate. And these data are from a paper that was just accepted. We got official notice today from Malaria Journal. And this is from a study that we're doing in Uganda. So let me walk you through how we compute the entomological inoculation rate. First, you need to catch mosquitoes as they're trying to bite humans, and there are a number of methods for doing this. I won't go into all of them, but one is the standard CDC light trap. In our case, we put a light trap like this next to a human sleeping under a net. So mosquito flies up, gets blown into the net and trapped. And you catch those and collect those and sort them out, so you've got all the anophelines. And the number of mosquitoes that are caught in that trap is plotted here. So we've got 100 households where we've got traps out, and this is a histogram. And what you're seeing here is a highly inequitable distribution of mosquitoes. So this one house in one night had 800 mosquitoes caught. and some of the other houses had not so many. So this is the 20/80 rule. Then you take the mosquitoes after you've sorted them and you test them to see whether they have sporozoites in their salivary glands. So this thick black line is showing the proportion over time that were positive for sporozoites. And you can see there's a bit of a seasonal signal, and this median here has been plotted on this red line, and you can see that there's they're going in opposite directions. When you have high mosquito counts, you tend to have low sporozoite rates. And that's probably because the mosquito populations are slightly younger because they're growing, and so they haven't had enough of them live a long time to become infectious. Anyway, the data look like this, and you take the product of those two things, and it's a measure of the number of, it's supposed to be the number of infectious bites per person per night. It's called EIR, the Entomological Inoculation Rate, and again, it's highly inequitable, highly heterogeneous, in fact, more heterogeneous than these by lots of measures. So what you're seeing is that even though the numbers aren't as high, this one house, there were 30 infectious mosquitoes in one night. So, and yet a lot of these houses had 0. So no infectious mosquitoes in a night. And I'm going to come back to that in a minute. If you were to take these two metrics, all the studies that have been done in the whole history of the world up until, we updated it in 2010. So the collection we have, this is a 2005 study, but we updated it 5 years later. What you're seeing here plotted on the x-axis is the annual entomological inoculation rate. So you take that measure and you extrapolate it up to a year. So this is 700 infectious bites per person per year at the right-hand side and 3 infectious bites per decade. So obviously not terribly reliable as you get down to that end. And the prevalence, the parasite rate, the proportion of people with malaria, actually proportion of young children, is plotted on the x-axis. And you see that there's a big cloud of points. There's about 120 of them. Now, if you wanted to, you could just take this gray line. It's a statistical relationship, and you find that they're correlated. That's great. But what we wanted to do is take some of those mathematical models and fit them to this data and see whether any of them did a good job. Well, it turns out one of them did a remarkably good job with only 2 assumptions, and only one of them was really necessary. And that's this blue line, right? This blue line is a model that says 20% of the people get 80% of the bites. You actually model a heterogeneous distribution of exposure. And when you do that, and in fact, we didn't tell it 20%, 80%. We fitted the parameter that would give us that, and it came out to 20/80. So that was good. And the other parameter that we fitted was one that tells us about how long an infection lasts, and that came out to be about 200 days, which is actually about the mean of the distribution from all of those experimental infections. So we didn't set out to pick those parameters. But if we had just said, well, let's pick an 80-20 rule and use the data, we would have got exactly the same numbers. So this has given us some indication that we've finally come to a model that does an actually pretty good job of explaining the relationship between EIR and PR. And we can use that relationship. In fact, we did use that relationship to transform the PR map into an EIR map. So now we have a measure of exposure that we can start to use in populations in a kind of an average way. And this has been in high demand from modelers and people all over the world. So this has been highly used. The problem is, so this heterogeneous biting model was disputed by some people because they said, well, the same effect could be coming out because of immunity. So obviously there was an identifiability problem that we couldn't get around using that data. So we went and we looked for another data set, and we started using this other metric, which is the force of infection. And actually, mostly this is derived from attack rates. So the fraction of an uninfected population that becomes infected in the next 2 weeks, and usually following these populations longitudinally. And it turns out as you increase the annual EIR in these studies over that same range, actually goes a little higher here, what you find is the force of infection, as measured by these attack rates, saturates. So these people are getting 1,000 infectious bites in a year, but they're only getting around 10 or 15 infections in a year. There's a huge disparity between those two numbers. And that's a pretty robust pattern. What I'm showing you, and you can read the details in this, is a review of all the studies we could find here, again, of every sort. And every color is a different study design, and every line is a different fit to all those aggregated studies. And you're seeing there are big differences, and yet they seem to lie right on top of each other. And some of these are actually really good studies, and some of them are really crappy studies. We're showing you all of them. And they all have the same basic pattern, right? Things saturate. Well, how do you get that? An 80-20 rule, right? these data that we're showing you down here are exactly the sort of data that would give you that pattern. And this is data from the field, not concocted from a model. So it's worth asking this question, why do we have heterogeneous biting? Why do some people drink more alcohol than others? Sorry, that's not what I was going to ask. Just seeing if you're paying attention. Why do some people get, well, we know some of the reasons from other studies. Proximity to larval habitat. So if you live close to where mosquitoes breed, you're going to get bitten a lot. If your house is open, has open eaves, that's another bad sign. If you don't use malaria control, so you're not using nets or area repellents or something, that's bad. There are clearly some elements of breath and sweat that are attractive to mosquitoes. Body size, dirty linens, having socks that smell like Limburger cheese. And what's the bottom one down there? Oh yeah, alcohol consumption. So all of those things will actually get you bitten by mosquitoes. And the fact is all of these are probably contributing to some degree. But from my perspective as someone who models transmission, what really matters is that they're heterogeneous, okay? Because that makes the models work. And so I want to come back to this concept of a reproductive number now, which describes the number of cases per case. So it's from a human index case to other humans, so it's the number of cases per case, or from an infected mosquito to other mosquitoes. And the basic reproductive number describes transmission under conditions where there's no control and no reservoir of malaria parasites and no immunity. So it's this highly concocted situation that doesn't exist anywhere in the world. And we also care about these other numbers, and let me explain them. If I take the same conditions as R0, but I allow there to be some control, that's also a relevant number. I'm going to call that control reproductive number. And then finally, there's this effective reproductive number, which I'm going to skip over to a large extent, which describes transmission in real populations. So it's a generalized concept with immunity, endemic malaria, and control. One of the things that you need to know about all of this, though, is there's this number we call kappa. And this was invented by two Toms and a Jerry. So this is Tom Churcher paper, and I'll walk you through this. And the other one was Tom Smith and Jerry Colleen. I've done a lot of the collection of data to analyze all this pattern. And the real question we have here is what fraction of people become infected, what fraction of mosquitoes become infected after feeding on a human, any human. So if you have a distribution of gametocyte densities in a population, and remember that's that graph I showed you before, and you look at how infectious you are at that level of gametocyte densities, and then you look at how often that person is being bitten, you can come up with a formula that tells you on average, at some point in time, what the probability is a mosquito will become infected. So what Tom Churcher and all those guys did is collected all the data on gametocyte densities, and they fitted these models, and what they found was the following. There is, like the parasites curves I showed you, so I was showing you prevalence of malaria, there's a prevalence of gametocytes that looks the same. And what's driving that is a decline in parasite densities later in life. And that's what they're showing you here. As you get older, you have fewer gametocytes. But because of the shape of this curve, you're only a little less infectious, right? There's still enough to infect humans a lot of the time. So we can actually define something called an effect size of immunity. It's the slowing of transmission by immunity, largely by the suppression of gametocyte production, and it's actually not that big a number, in my opinion. Now this is, I'm gonna be really honest with you, there's a disagreement about how important this number is out there and how big it is. I am of the opinion, especially after papers like this, that it's on the order of 2 or 3. But some other people believe it's bigger, say on an effect size of 10. So it's slowing transmission by 90% or so. But for now, take my word for it, it's small, 2 or 3, and I'll show you exactly what I mean by small in a bit. Small certainly relative to EIR variability. So we know then that our R0 is related to something about transmission in a population, and it turns out, since R0 depends so much on vectorial capacity, that I can actually measure vectorial capacity from EIR if I know something about kappa, that number I just told you about. So we use this as a basis for inventing a formula that derives R0 from these estimates of EIR. So here is our range up to 700 infectious bites per year. These are under 2 different models. And we're saying that R0 is a number that ranges up above 1,000, in some cases up to 10,000. Okay, this is really bad news, right? Because R0 is this measure of how intensive transmission is, and if it's really that high, it means you have to get a 10,000-fold reduction in transmission to eliminate malaria. So I'll come back to that in a bit, but what I really just wanted to show you is we can use that relationship now, and we have developed this global map of reproductive numbers. Now, it's reproductive numbers under ambient level of control is my one caveat, but I'll come back to that too. So now we have an order of magnitude, likely over 100, probably as high as 10 or 1,000 or 10,000 in some of these areas in Western Africa and northern Mozambique. Okay, so transmission of malaria can be very, very intense. So I'm going to switch gears just a bit and then come back to control. So the other part of transmission is how the parasites move around, right? If there is heterogeneous biting, we ought to be able to find those pockets and attack them. So this is a paper where we talk about hotspots. So this is more of a sort of a call to arms. We ought to go out and look at distributions of malaria and find out how heterogeneous they are. Here's some real data. And this is a paper that's coming out in eLife. So what we're looking at is DSS data, and this is the fraction of the fevers in a household that were malaria positive, right? So it's kind of an odd statistic, but it's the only way you're going to get sort of extensive and detailed data on the incidence of malaria in a population over very long periods of time. And what I want to show you is in this, several thousand houses, there are big pockets where there's no malaria. There are little pockets where there's no malaria. There are big pockets where there's lots of malaria, and there are small pockets where there's malaria. So we actually did the analysis, and it turns out that, so this is an old idea, apologize, but we revived this idea of fractal, of self-similar distributions. And it turns out there are hot spots everywhere, and there are hot spots within hot spots. So this is a paper that's just been accepted at, well, it's in review, but likely to be accepted at eLife. So we wanted to get an idea on how that happened, and we built some models, and I'm not going to tell you about them because I'm running out of time. But one of the questions we really, really need to know is what are the spatial scales that characterize transmission? So this got us into some other things. How do mosquitoes move around, and especially how do humans move around? Now, when you think about humans moving around, you sort of, how far do they move is on the x-axis, and how often do they move that far on the y-axis? And you can think about all sorts of causes for why people move around. So this has been a big part of what we've been doing, is assembling and analyzing these data lately. So here is analysis of the way people move around Iquitos. This was largely for a study of dengue transmission. This is a study that we did in Kenya, where each one of these dots is a cell phone tower. There's all the cell phone towers. And we have call data records from all of the cell phone owners from a particular company who shall remain unnamed, but they have about 90% of the market. And this, so these sort of different colors are the natural clusters that come out of this, and the background is the prevalence of malaria. So red is very intensive transmission, and yellow is not very intensive. And by the way, down here in the corner, you can see that same slice. So that little bit of Kilifi is right down there in that one pocket of Kenya. Now you imagine you have to scale that up to the whole thing. So what we can do is analyze this and actually look at the way parasites are flowing around landscapes. And this is really kind of a critical idea. There's a lot of transmission around the lake and a lot of movement from the lake to Nairobi. There's not very much transmission in Nairobi and a lot of people moving back and forth. So what you get here is the way people move around, and there are kind of sources and sinks of movement of people. And of course, Nairobi is a big sink for moving of people. More people go to Nairobi than any place else. But there's also these huge source-sink relationships in the movement of parasites. And that's sort of weighted by the prevalence of the parasites. And you see there's this dominant, this is a dominant source and this is a dominant sink. And so you see this, you can actually now track the way parasites are moving around. Well, we did this also at a continental level, looking at malaria communities. These are areas of Africa that are more closely related to one another than they are to sort of surrounding areas. And you see these colors as you start to glom these things together, unite until you get a picture that looks like this and seems to make a lot of sense. There's a community in West Africa, East and Southern Africa, this sort of Southwest Africa, middle, and so on. These things at the end. But really what you're describing is a complex community structure, and the underlying data here were census data, number of people from country X living in country Y. And all right, so what are we going to do with all of this? Well, now we need to actually come back to this idea of hotspots, right? So we have the data. This just came out a week or two ago. This on the top is a high-risk area for malaria in Namibia. What you see, though, is if you start looking at how people are moving around Namibia, you can identify sources and sinks even within those hot and cold areas. And so, depending on how much you want to do targeting, you can reduce this population that you have to worry about from 1.3 million down to a much smaller subset of about 0.2 million. So we're talking about a six-fold increases in your cost. And what we're positing here is that if you actually targeted these populations, these red ones here, you would get a much bigger bang for your buck in all of the places that are surrounding it. So now we've turned, abstract ideas and data into pragmatic advice that will be used by Namibia in the way they design their malaria control. The question is, how do we evaluate that to say whether we were right or wrong? I mean, everybody's been clamoring for this stuff, and we did it. We don't know whether we're right or wrong. We think we are. It would be really nice to know. So we have to do the evaluation component of this, and that's where we are. So a theory of control. Now, I'm going to try to go through this as fast as I can, but it's very critical. When George MacDonald in the 1950s was involved in all these debates about global malaria eradication program, it was because of DDT. DDT was this new miracle tool for malaria control. You paint it on the side, you spray it on the sides of a wall, a mosquito would land and die. And he showed in this analysis in a paper in 1952 that the reason why DDT was so effective is that it prevented mosquitoes from living long enough to become infectious, and then limited the number of bites they gave afterwards. And the way he summarized that was in this table, where on the sort of columns you're showing probability of surviving X days for various values of P across the top, right? And so the message was, if you use something that killed adult mosquitoes, it shortened their lifespan, it had a really big effect. So you can see that in this picture here. So this is the proportion of an original cohort that is alive and infected, in the dark purple, or alive and infectious, under certain assumptions about how frequently they're becoming infected. Now, if you go to that population and you put in nets, or indoor residual spraying, and you shorten the lifespan, now this lighter purple is giving you a smaller proportion later on that are alive and infected, and this little pink notion down there is a proportion that were alive and infectious. Now, what he's saying here is that you get a big bang for your buck by going after adult mosquitoes. If you could kill them faster, that would be a very, very good thing. Now, this is good advice generally, but I think it's goofed us up in some ways because, well, if that's what you do, then you always have to go out and kill adult mosquitoes. And that's the answer that's been given over and over again, even in places where it might not be appropriate. So what's wrong with that? So to get at what's wrong with that, we wanted to come up with a better notion, a more general notion, and this is what we're going to call an effect size. An effect size is a proportional reduction in transmission in the same units of R naught. So if R naught were 10, you have to achieve effect size of 10 to achieve elimination. We put it in the same unit, so it'd be really easy, okay? It's as good as it gets. But the main thing is that effect size is related to your coverage level. If 50% of the population has nets, it's, coverage is somewhere lower than 50% because it's how many they're using. But if you got it up to 80% or 100%, then your effect size would probably be bigger too, right? So there's this relationship between an effect size and effective coverage that you want, all right? And we can do this for nets or spraying or anything we want. So we can talk about effect sizes of immunity, of transmission, and something I'm not going to talk about, or the reservoir. So I mentioned nets. So the way we come up with these guesses about effect sizes, we model the way mosquitoes behave around nets, whether they bite a non-human host under some conditions, whether they go into a house with a net or not, whether they bite a person under a net or not, whether they die or not, and how many times they repeat this. And we come up with a measure of how vectorial capacity is changed by the presence of a net. And we get this relationship between effective coverage and an effect size, right? And the thing about this that I want to point out is effective coverage is on a 0 to 1 scale. It's a proportion of something. The effect sizes is on a log scale, right? Effect sizes get very, very big as you approach 100% coverage. It's just the way things go with vector control, and it's partly due to that killing effect of adult mosquitoes. We have also now, for the very first time in the history of the world, developed models for the effect size of larval source management and of routine medical treatment. I'm not going to go into them because we're short on time. I'm also going to have to skip this. So here's the way we actually use those nets. So the problem is everything we do, we want to know what R naught is. But we don't know what R naught is because in that population there's some level of coverage. So we can actually get around that in a very clever way. We can actually set marginal coverage targets. So we pretend that if a population has 50% covered, that they don't exist, and we set coverage targets on the remaining 50%. And this turns out to work extraordinarily well so that we can now go into a population and tell people how much they have to improve their current use of nets or drugs in order to get things done. So here, for example, we tell you where elimination would be possible in the world today if you could just get up to 90% of the people who are currently not being treated with drugs treated. So just by improving your medical system, now it's not easy, I'm not saying that, but we have a basis now for setting intervention coverage targets and especially marginal targets. Now, effect sizes are so incredibly useful because we can put anything onto this scale that affects transmission. Global warming, yep. Immunity, yep. Interventions, yep. Economic development, yep. Okay, and so we did. What you see at the top here is a map of malaria that was produced in 1969, and it is putatively malaria distribution in 1900. This is called the Lysenko map. This is the map we produced in 2007 in 2009 describing malaria in 2007. And what you're seeing is that between 1900 and 2007, there's been a massive contraction in the range of malaria. It's contracted. Many people who used to be at risk are no longer at risk, and this is the dominant pattern you're seeing across the world. Now, it's oversimplifying a complicated story, but we can associate effect sizes. We wrote this in part to answer concerns about global warming and malaria. The answer was, yes, warming has an effect size, but it's puny compared to everything else that we do. Don't worry about it. If you want to actually take care of malaria, you have the tools you need to take care of malaria. And that was our message. So we can worry about insecticide resistance and drug resistance on the same scales. And again, so here's the theory of elimination in a nutshell. I have a basic reproductive number. I have a potential intensity of transmission at some level of effective coverage. That defines an effect size. So to eliminate malaria, I have to get an effect size that exceeds R naught. So I come into a population, I do intensive control, I suppress transmission. The parasite reservoir starts to decline, and I have to keep that up until the parasites are gone away. And then the question is, can I let the foot off the gas pedal, right? Or off the brakes? Can I do something? Elimination requires sustained management of imported malaria. So in the United States, for example, we've had about 1,000 cases of malaria, about 1,400 cases every year. They don't spread, and the reasons why are of interest, but I'm going to get into that. So here's what happens typically in places that don't eliminate malaria if you do take your foot off the gas pedal. So this is a problem we worked on in Zanzibar. Quickly, 1959, lots of malaria come in, global malaria eradication programs, spraying programs. Malaria drops like a rock. Prevalence is down around 2 or 3%. Malaria, the Sultan, who was ever in charge, said, well, Zanzibaris are somehow supernaturally now immune from malaria. You guys can go. They go, and malaria comes back, of course. And so in 1980, they formed the Zanzibar Malaria Control Program with some help from USAID. Malaria starts coming down. And then we have this problematic era where no one actually did a parasite survey that was extensive over the whole island, except for this one study right before things really started to change in Pemba and things come down. So what you're seeing overall is this period of do control, malaria goes down, stop control, malaria comes back, do control, malaria goes down, stop control, malaria comes back. And we actually documented it in this paper hundreds of times, or 79 times, right? And the main reason, there were lots of reasons that were blamed, and of course it's problematic because nobody's studying malaria when it comes back. If you were, there would be money being spent on it, and there was no money being spent on it. So people prognosticate, but the reason most often given was they weren't spending money. Well, here's the really interesting bit. Countries that did eliminate malaria saw a completely different pattern. And this is very significant. So this is 30 countries over 31 years who have reported in some public place where we could find it, the number of imported malaria cases and the number of locally transmitted cases. So the way I'm showing you this is the size of the square. The key is here. So that's 10,000, 1,000. You're seeing the number of cases. So France is 2,000 or 3,000 cases of imported malaria each year. And the color is telling you something about the number of transmitted cases. So you're seeing a lot of white here, meaning there was a lot of imported malaria and no onwards transmission. And if you were to sum this up, there are about 250,000 imported cases documented, and only about 5,000 transmitted locally, which is a very, very low reproductive number. Malaria elimination here is incredibly stable. And this is important, right? Because if you can go into a place and eliminate malaria and then take your foot off the brake and not have it come back, then you have a basis for planning malaria eradication at the whole globe, right? So the question now is, why did this pattern occur? And these papers, we actually discussed some of those reasons, but one of them may be the health system. So that paper I skipped over about drugs, one of the messages is if you could treat everybody promptly with drugs, you would end transmission. Well, not end it exactly, but one of the quirky details about that gametocyte production is gametocytes start to get made probably day 7 or 8. Parasites come out of the liver day 6, made about day 7 or 8, take 12 days to mature. A person is not infectious with malaria until about day 20 of an infection. So if you treat them immediately and promptly and end all of that gametocyte production, and even with a gametocytic drug, you can stop them from ever transmitting. So in a good healthcare system, you need not ever have an outbreak. A good health system, of course, is one where everyone gets treated. So this is a kind of a complicated picture describing how that works, but as clinical immunity wanes, so people with clinical immunity don't get treated as often, things become more dangerous, but they also become easier to control. And that makes it possible that you can get rid of malaria. Dang, I'm not going to get through all of this. Well, let me go to where we are at the end today, right? We're going to have to understand, using maps like this, which describe the vectors. I'm not an author on this paper, by the way, that this is a sort of dominant vectors of malaria across the world. And there are lots of them, and they have unique behaviors in each of these, and probably even within their ranges, different behaviors and biting preferences. And we have to use maps like this to make plans, to go set targets. And we have to combine them with maps like this, which tell us something about the overall intensity of transmission, to tell us about how operationally efficient we have to be, right? And whether we can take, bits of that have color. So that's where malaria is today. And can we turn them into bits that are gray, where there's no malaria, that will be stable and not have malaria elimination at all anymore in the future? That's the problem that we've set for ourselves right now, because if we could do that and we could come up with a plan for transforming the world bit by bit from malaria endemic to malaria free, then we would eventually get to eradication. So can we find areas that are large enough that there's essentially no malaria coming in or very little coming in, but they're small enough to tackle with an intensive program with existing resources? Let me just conclude, and then we can go to a reception or ask questions or whatever. So maps and models are useful tools, but they're more useful when they're combined. We can do this to do things like strategic planning or estimating the avertable global burden of malaria. The way forward is to evaluate the interplay between not just the technical. So those coverage targets that I told you about depend on the vector, they depend on the people, they depend on the operations. So if I tell you just got to go kill adult mosquitoes, it's just not useful. You have to know what it is that, what are your vectors? Are they actually, are you going to reach your targets by just trying to kill adult vectors with indoor residual spraying? We need to actually look at the interplay between the technical requirements for achieving some policy goal and the operational constraints. And if we don't, then we're just going to keep on, bumping our head against the same old problem. I'm not saying it's easy, but I do think it can be done. So we need to look at this, come up with a framework, and a lot of what I skipped over is how to get this optimal interventions in context, to develop these stable malaria-free zones so that we can convert the world bit by bit to being malaria-free. And the one punchline that I am going to commit myself to is that for that to work, for that plan to actually do, we're going to have to invest in making sure that the health systems, which are going to be the places that stop these outbreaks in the end and keep them malaria-free, have to be highly functioning. And so maybe now for a lot of these endemic areas, and this is going to sound heresy, but nets are maybe not the best thing to be doing. Because if you could get the drugs out, you could save more lives and not spend as much money. Meanwhile, getting the health system to a point where then you bring in the nets and malaria goes away for good. I think it's a much better plan. People are not going to like me for it. But the other thing, so here's the end. I have a lot of people to thank because I talked about a lot of papers and didn't talk about a lot of papers. The blue papers, by the way, are all things that were published since I got to Hopkins. So I just wanted to point that out. I also wanted to say thanks to all of the authors on all of this paper, and it is a substantial fraction of the mosquito-borne disease modeling world and the mosquito malaria epidemiology world who participated in these papers and others like them. So that's it.
Okay, so I'll ask people to use the microphones if they're going to ask a question. Let me just start. So in areas where malaria has been eliminated, is there change in the distribution of the vector?
So the question is about, I think, generally, what were the causes of elimination? So one story that I really like about changes in the vector was not about their distribution, but about their behavior. In Europe, a lot of areas, and this is before DDT or anything, spontaneously became malaria-free. And a study by a guy named Hackett in the 1930s went back and actually discovered that the behavior had changed. The housing quality had improved, and mosquitoes had stopped biting people indoors. Mosquitoes are spending more of their time in barns biting. They'd become essentially zoophilic, and they weren't biting humans anymore. The areas that still had malaria, by and large, still had a lot of anthropophilic vectors. So even without changes in the vector distributions, you could get big changes in the vector behaviors that end malaria transmission, and that did happen. I'll tell you another anecdote from Uganda. Some friends of ours were looking at, in Kampala, they were looking at community gardens and the role they play in malaria transmission. And they came back for their next field season, and the garden was gone because they'd built a factory on top of it. So was malaria. And so Kampala is now, I don't take my prophylaxis when I go to Kampala anymore. And I don't know what's changed, but it's probably the vectors are gone.
Questions, comments? So maybe, Clive, I'll pass it to you first, and Josh, can you bring the...
That's great. It's good to get a picture of of all your thoughts put together. But one of the things that concerns me as we're finding in Zambia now is that a large proportion of the malaria cases are actually asymptomatic. Probably 70% of malaria is asymptomatic. And so it's very hard to, whereas these do come into your model, it's very hard to find a way of actually getting at them to get rid of the parasite.
So yeah, let me just say that this curve, which it's something that makes mathematicians very happy, but takes a long time. So describing the phenomena that you're saying, if you have endemic malaria in a place with clinical malaria, it may be very hard to get rid of it. But ironically, then in those same places, once you've gotten rid of it, it might be very hard for malaria to reestablish. So it's this kind of weird story that malaria can be very, very hard to get rid of, but once you get over that hump and get rid of it and immunity wanes, then it doesn't come back. And that, I don't think that applies everywhere, but it might apply to large chunks of Zambia, especially where you work in Macha.
So when you say get rid of, right, so did Zanzibar get rid of it? It was very low, it looked like.
Right now, the prevalence of malaria in Zanzibar is below 1%, and we don't know whether I haven't been staying in touch with it. We don't think there are very many endemic pockets. So the story now that most people believe is that the fishermen who live on coastal Kenya and Tanzania and go back and forth, some of them are polygamous and have families on both sides, that they're a big part of the reservoir that's reintroducing malaria continuously. Now that's not supported by anything, but the places where they keep having problems are either the old rice paddies, which would suggest endemic transmission, or places where fishermen come ashore, which is actually much of Zanzibar.
But back, you showed the history of, when there was a control program and it got very low in Zanzibar in the 60s, I think. So
yeah, and now too.
Yeah.
Now it's as low as it's ever been.
Yeah. So when you say, get rid of, what number?
0. So right now Swaziland is at a place where almost all of its malaria can be traced back to cases imported from Mozambique. So when you get to those end phases, you switch really from, and you are chasing down individual cases, and many of them are asymptomatic, but when you do find them and you ask them, you know, have you been outside of Zanzibar, or sorry, have you been outside of Swaziland? More than half of them, like 70 or 80% of them, have been working in an area at risk recently in Mozambique, and the other 20 or 30% not. So when you do the calculation, the idea is that most of the local transmission is stopped. What you're seeing is like little, you know, chattering chains that are stuttering out. They're not lasting very long, and those short transmission chains are, you know, constantly being reintroduced. So when I say zero, I mean that there are no local chains persisting. So there's nothing left from the days before. That's all gone, and what you have left is all coming in from outside of some artificial circle, admittedly, that you've drawn, but an artificial circle that you've drawn.
Yeah, hi. Great talk, thanks very much. I want to go to your last point, which is improving health systems. So saying if we could only make health systems work, we could eradicate malaria is kind of like saying if we could only put a bell on the cat, then it wouldn't catch mice anymore, or if we could only eliminate poverty, then nobody would be sick. What is it that makes a health system work in a sustained way? Have you looked at changes in health systems in Europe or other places like that that have eliminated malaria and figured out what happened differently than is happening in malaria endemic countries right now, where we've invested billions of dollars in health systems over the years, and we're not doing much in terms of system strengthening because we tend to like vertical programs because they produce more rapid results that make donors happy. But even where we've tried to do health system strengthening, it doesn't work very well because it's hard to keep track of where the money goes, and there are no indicators that tell you very effectively what's happening, and we don't seem to be getting anywhere. So how is it that we are going to make all these health systems work and make sure people can get treated in an opportune way?
The performance measure that I care about is what fraction of incident malaria cases get treated properly. And there's a very simple measure for it. So how are we going to get there? Well, one thing I think is that it's going to happen on its own. So, you know, the world of tomorrow is not like the world of today. If you look at projected demographic, health, and wealth data into the future, Africa is going to start looking like a lot of the rest of the world 20 or 30 years from now. It's going to take 20 or 30 years, and there will be problems. But right now, childhood mortality rates in Africa are falling. Wealth is rising, and when people get wealthier, health is one of the things they buy. So this transformation is going to start happening in various places on its own. And my contention is that, you know, that is going to be our ally, and we can push it forward faster by doing smart things like vertical programs which have a component of health strengthening in them. So the diagonal idea, right? You do a vertical program, but it's supposed to have some health system strengthening component of it that produces these horizontal benefits, and that could help spur you along. I mean, this was the argument I heard about polio eradication, the vaccination programs having a benefit throughout South America, and that it wasn't polio that was a big recipient, it was the strengthening of the health systems that benefited from having a polio vaccination program in place. So I think there is a value to a vertical program even in those systems because it brings money in and specific performance measures, but you have to be cognizant of the fact that if you don't strengthen those health systems in that horizontal way, you're not going to get there in the end. I would also say it's not going to happen on its own if you don't have a population that's, you know, at a point where it's about ready to change. So, you know, there are some areas of, you know, very remote Odisha in India where you still can't get the doctors to get them to give the right drugs. I mean, they've got ACTs, they're in the clinics, and they're not using them appropriately. Whether that's a matter of wealth or culture or something else, I don't know, but, you know, I don't have an answer for it. I just said that has to change before you can actually go in and stably eliminate malaria.
The last question, Ken.
I was intrigued by your comments about how mosquitoes adapted to humans or non-humans or animals, and so I always had the the thought that a mosquito was genetically programmed on what it was going to bite and which blood it would take and which egg it would lay, and et cetera. But how much do, let's say, the same species of mosquito change given the environment, the behavior? Because that's another avenue, I guess, of control, of understanding the disease, at least.
I think in some populations there's genetic variability for resting and biting preferences that could be selected to help or hurt us. And so you can get a change in behaviors. And I'm not even talking about, physiological resistance to insecticides, but just changes in the way mosquitoes behave that makes it easier or harder to control or easier or harder for them to transmit. The extent to which that variability exists in the main vectors that we care about in Africa, the ones that are causing, all the African kids to die, I don't. My impression is that in Anopheles gambius entu stricti, there's not a whole lot of variability in that. I mean, they like humans, and they don't do much but munch on humans. So in Arabiensis, I think there's more. That's a sister taxa. There's more variability, but that ends up hurting us more than helping us because then they turn to cattle and still opportunistically bite humans. So our nets and our other tools don't work quite as well. It's a complicated question, and I've talked about 2 out of maybe 40 or 50 vector species across the world that we would have to go into in extensive populations that have variability in them. So it's a fun topic, but Doug Norris is going to answer all your questions later.
So maybe out by the water.
Yeah.